All right, guys, let's get started with part two of lesson one. Today, we're going to learn about microscopes. Our success criteria for today. So, by the end of this lesson, you should be able to describe how microscopes produce magnified images. Okay, so do you remember from last time the cell theory? It states that all living things are composed of cells. It also states that cells are the basic units of structure and function in living things. And it also states that all cells are produced from other cells. This theory is super important to remember as we move through the rest of the unit. So you might want to write this down again. Um, Okay, so the cell theory could not have been developed without microscopes. Some microscopes focus light through lenses to produce a magnified image, and other microscopes use beams of electrons. Both light microscopes and electron microscopes do the same job in different ways. For a microscope to be useful, it must combine two important properties: magnification and resolution. Have you ever looked at something through spilled drops of water? If so, did the object appear larger? Magnification is the condition of things appearing larger than they are. Looking through a magnification glass has the same results. A magnifying glass consists of a convex lens which has a center that is thicker than its edge. When light passes through a convex lens and into your eyes, the image you see is magnified. Magnification changes how you see the object and reveals details you may not have known were there. Take a look at these four pictures. All of these images have been magnified, which makes them look unfamiliar. I want you to, in your notebook, number 1, 2, 3, 4, and write down what you think each of these pictures are, and explain why you think that picture is showing what you think it shows. This here is a compound microscope. It's a type of light microscope that we will be using in the classroom. This type of instrument magnifies the image using two lenses at once. One lens is fixed in the eyepiece, and a second lens is chosen from a group of 2 or 3 lenses on the revolving nosepiece. Each of these lenses has a different magnifying power. By turning the nosepiece, you can select the lens you want. A glass slide on the stage holds the object to be viewed. A compound microscope can magnify an object more than a single lens can. Light from a lamp or reflecting off of a mirror passes through the object on the slide, the lower lens, and then the lens in the eyepiece. The total magnification of the object equals the magnifications of the two lenses multiplied together. For example, suppose the lower lens magnifies the object 10 times, and the eyepiece lens also magnifies the object 10 times. The total magnification of the microscope is 10 times 10, or 100 times, which is written as 100x. Excuse me. I know that that's a lot, so if you need to pause, rewind, and listen to that again, go ahead, because we're going to actually calculate that on the next slide. So this microscope, which is a compound microscope that we'll be using in our classroom, has a 10x lens in the eyepiece. The revolving nose piece holds 3 different lenses, a 4x, a 10x, and a 40x. So in your notebook, calculate the 3 total magnifications possible for the microscope. And remember, you just multiply them together. After that, answer the question, also in your notebook. What would happen if the object on the slide were too thick for light to pass through it? So what do you think would happen if the object that we were trying to look at on the microscope was too thick for light to pass through it? You can pause. Okay, one more thing. Electron microscopes. The microscopes used by Hooke, Leeuwenhoek, and other early researchers were all light microscopes. So those are the guys that we talked about the last couple days. They were the early pioneers of microscopes. All of those were light microscopes. But since the 1930s, scientists have developed several types of electron microscopes. Electron microscopes use a beam of electrons instead of light to produce a magnified image. Electrons are tiny particles that are smaller than atoms, just in case you didn't know that. By using an electron microscope, scientists can obtain pictures of objects that are too small to be seen with light microscopes. Electron microscopes allow higher magnification and better resolution than light microscopes. Okay, so to review these key concepts, I want you to answer the following questions in your notebook.